Welcome to the Development in Snails module. In this talk, we're going to learn about the specification process in snails. We're now going to look at the fate maps in snails, and they can be determined by injecting fluorescent molecules in specific micromeres of snail embryos. Now, the fluorescence can then be observed as the embryo undergoes further development, and that is what is shown in the figure. So, let us look at the micromere 1b. So if we add a fluorescent dye to the micromere 1b, which in this figure is also called as the blastomere 1b, we can observe the fluorescence in the 2-day-old embryo, and we can see how the fluorescence changes, which associates with more and more daughter cells being made and then moving to different portions. So we can see that how in the 3-day-old there is a change in the cell migration pattern, and then we can observe it until we get the larval stages. And so by using these kinds of experiments, fate maps have been developed for snails. Now based on the fate map, what we know now is the first quartet of micromeres, which are the 1A, 1D to 1D micromeres, and some cells that are present in the second and the third quartet form the head ectoderm. And that is what is shown in the blue box. So in the embryo in the blue box, we can see the first quartet micromeres, which are 1A to 1D, and they all have been shown to form the head ectoderm. On the other hand, when we look at other parts, we know that the first and the second quartet cells develop into the nervous system. And the macromeres A, B, C, and D can become the endoderm. When it comes to the mesoderm, you can have the second and third quartets forming the musculature, whereas organs that arise from the mesoderm, like the kidney, heart, primordial germ cells, or retractor muscles, are formed from the 4D blastomere, which can also be called as a micromere. All of this is shown in the figure in the blue box, where we can see which cells provide which germ layers in the embryo. Now in the case of snails, cell specification is determined by morphogenetic determinants that are present in specific regions of the oocyte. So we already saw the fate maps, which tell us which cells will form what kind of parts of the organism, and so we know which specifications occur in different parts of the embryo. Now we're going to see how the specification is brought about. Now in many cases, the mRNA for some transcription factors and paracrine factors, they actually associate with one of the centrosomes that is present in the cells. This centrosome association will allow the mRNA to enter into one of the two daughter cells. So it should be noted that the mRNA are associating with only one of the centrosome. Now do remember, during mitosis, the centrosomes will undergo duplication to form two centrosomes. Now the messenger RNA are present with only one of the centrosomes, and that is why they can enter one of the daughter cells and not the other. This is what is shown in this figure, where let us look at this 4-cell phase where we're going to have the mRNA of a transcription factor or a paracrine factor that is shown in green. And it likes to associate with the centrosome that is shown in blue. So in this case, the mRNA is present in all 4 cells, and it is associated with the centrosome. Now when this 4-cell embryo undergoes mitosis to form the 8-cell embryo, what we can see is we can observe that now the messenger RNA is present in the micromeres and not in the macromeres of the embryo. This happened because when mitosis happened, that messenger RNA were associated with the centrosome, and that centrosome goes to the daughter cells that will form the micromeres. Now when this 8-cell embryo undergoes another round of mitosis to form the 16-cell embryo, once again, because of the centrosome association, only certain daughter cells will get that messenger RNA. And this way, we are able to have morphogenetic determinations present in specific places of the developing embryo. Finally, in this figure, when we look at the 24 cell stage, we can see that messenger RNA is present only in certain cells. And when that messenger RNA gets translated into proteins, those proteins are present only in certain types of cells and can determine their cell fate in this manner. We're also going to look at another feature that happens during the embryogenesis, and this is a structure that's called as the polar lobe. Now, before the first cleavage event happens, a ball of cytoplasm is formed that is called as the polar lobe. In the figure that is shown below, what we see is the part in the blue box, which is the polar lobe. The polar lobe only has cytoplasm in it. There are no nuclei in the polar lobe. Once the first cleavage occurs, the polar lobe gets associated with the CD blastomere. So when the first cleavage event happens, we get two cells, the AB cell and the CD cell. And that polar lobe gets associated with the CD cell and not the AB cell. The polar lobe provides a three-cell appearance. So when we look at the picture in the blue box, it seems like as if there are three cells. However, only two of these have nuclei and the third one is the polar lobe. And so just because it shows a three-cell appearance, the embryo in this stage is called as the trefoil stage. Now, if the polar lobe is removed, the development can continue, but the larvae that are formed show incomplete development. And so that way it is now thought that the polar lobe will have certain mRNAs and proteins that play an important role in development. So we saw the role of the polar lobe in the first cleavage step. Now let's see what happens in the second cleavage step. So before the second cleavage, the polar lobe is formed now in the CD blastomere. So after second cleavage, the polar lobe gets associated with the D macromere. And that is what is shown in the figure below, where we have our AB and CD blastomeres. The polar lobe develops in the CD blastomere, and when the cells undergo cell division to form the second cleavage event, we end up with four macromeres, A, B, C, D, and of these four macromeres, the polar body is present in only one of them, which is the D macromere. This is one of the reasons that the D macromere is the largest, because it has all the excess cytoplasm that comes from the polar lobe. Now, the polar lobe contains morphogenetic determinants that are responsible for forming heart and intestines. They also contain factors for inducing the development of other cells, and that's why removing the polar lobe will cause developmental defects. Since the polar lobe is associated with the D blastomere, if we remove the D, 1D, or 2D blastomere, the larvae that develop lack the heart, intestine, and other structures like the eye, foot, shell gland, and the velum. And this is what is seen in the figure. The figure to the left, which is the A figure, is normal development of the embryo, where all the blastomeres are intact. However, when we look at panel B, that is to the right, in this case, the blastomere has been removed, and the one that has been removed is the D blastomere. And because of this, there is incomplete development. Thus, the phenotype of the embryo where the D, 1D, or 2D blastomere has been removed shows a phenotype that is similar to when the polar lobe is removed. Now let us look a little bit more about the role of the D blastomere in development. If the macromere 3 capital D blastomere is removed, it is found that the larvae that develops lacks only the heart and the intestine, but has the other bodily structures. Now, if we let the 3 capital D blastomere undergo cell division, it gives rise to a capital 4D, which is a macromere, and the 4 small d, which is a micromere. And when we remove the capital 4D, or the macromere, it doesn't produce any changes in the development of the organism. On the other hand, if we remove the micromere 4D, then it results in a larva that does not have the heart or intestine. This shows that 4 small d blastomere has morphogenetic determinants that are required for the formation of the heart and the intestines. The 4 small d micromere is able to undergo cell division to form cells that will form the endoderm and the mesoderm, and hence it shows autonomous specification. It is known that beta-catenin and nanos are present in 4 small d, and they play important roles in the formation of the heart and intestines. Thus, the D blastomers play very important roles in snail development. When we look at the D macromere, it contains the determinants for proper cleavage. It also contains determinants for autonomous mesodermal and intestinal specification, as well as it enables inductive interactions that will lead to the formation of the shell gland and eye. With this, we come to the end of overall cleavage that happens in snails. With this, we come to the end of our talk, where we learned about fate maps in snails and how messenger RNA and paracrine factors are localized in specific daughter cells. We also learned about the role of the polar lobe and the D blastomeres in development.